from my side. I'm your host, Omar, and this is the first webinar of the NeuroMac webinar series. And today's guest is Dr. Kiavash Movahedi. Dr. Movahedi studied bioengineering in Brussels, and he followed then by a PhD, where he studied majorly the heterogeneity of myeloid cells in cancer. He has done some nice publications in that regard with tumor-associated macrophages in specific. Afterwards, he did a postdoc at the Max Planck Institute.
Thanks a lot, Omar. So I'm really happy to have received the invitation. This is actually my first webinar ever, so either as a participant or a speaker. So I'm quite excited to see how this will work out. And please ask as many questions as you want. I really like to have an interactive discussion. Okay.
We have, so just that you are aware, we are here in the room around 15 to 20 people. And I guess I don't know, they maybe want to say hi, and I'll just do it quick. I hope that they are clear. Yeah. Okay, doke.
Right. All right. So indeed, as Omar said, I'm from Brussels in Belgium, so from the VIB Center of Inflammation Research, where I lead a group that's mainly interested in neuroinflammation with a strong focus on brain macrophages. Okay, and when you typically talk about brain macrophages, people think of microglia, which are the tissue-resident phagocytes in the brain parenchyma. They have been quite a hot topic, right? Because we're now realizing that they're probably really important for disease and for homeostasis. So the last decade, we've actually learned really a lot about these cells. We now know that these are long-lived cells, that they are embryonically or yolk sac derived. We know that they self-renew in situ, right? They don't receive input from bone marrow. At least this is in laboratory mice, right? Because we don't know whether this is really true in humans. Also, we don't know whether after inflammation and resolution of inflammation, this would still be true. But anyway, for now, this is what we know. They're also highly dynamic cells. They're continuously surveying their environment. They're involved in synapse remodeling, in assisting neural circuit formation. And also, as I said, they are implicated in many neurological disorders. Okay. So, however, today I will mostly talk about the other brain macrophages, and these are the resident macrophages that are sitting in the non-parenchymal sites, which we call the border sites. Okay. And maybe first let's discuss a bit these border regions. So first of all, you have the three-layered meninges, which is sitting on top of the brain. This consists of the upper part is the dura mater, followed by the arachnoid and the pia mater, right? And these two, we also can call them the subdural meninges. Now, what is interesting is that the dura actually is an open barrier. So its vasculature doesn't have tight junctions. It actually is fenestrated, right? So meaning that it's easy access for cells and biologicals to move into the dura. Now, the barrier function is taken up by epithelial cells, which we call the arachnoid barrier cells, which do have tight junctions, and they're limiting the diffusion or the movement from cells from the dura into the subdural meninges. Now, in the subdural meninges, the vasculature does have tight junctions, so it's a real barrier, and it's called the blood leptomeningeal barrier. It's a bit different from the blood-brain barrier in the parenchyma, as for example, it's missing the astrocytic end feet. Okay, but in effect, it's a strong barrier system. Now, the brain also has these large holes, the ventricles, which are filled with cerebrospinal fluid, and we have the choroid plexus that is floating in these ventricles. Now, the choroid plexus is actually just a monolayer of epithelial cells, and in between that, you have a thin connective tissue that is called the stroma. The stroma is also highly vascularized, but again, the vasculature does not have tight junction. It is fenestrated, so again, it's an open barrier system. And once more, the barrier function is taken up by the epithelial cells, which again have tight junctions, and they limit migration from the stroma into the CSF. Now, it is actually thought that these epithelial barriers, especially in the choroid plexus, that they actually act more as a smart gate. So they're not as strict as, for example, the BBB. And so under certain conditions, they can open up or they can allow the movement of cells into the CSF. So it's more of a smart system, smart gate system. Now, as you can realize, so the fact that these are open systems means that there is probably an easier way to survey blood-derived molecules. So this is probably, these border regions are important for blood-to-brain communication. They're going to be very important for entry of immune cells into the brain, and probably just for orchestrating neuroinflammation. So as such, we're actually quite interested in studying these sites. And in the first instance, we wanted to really get a grasp of the immune complexity in these border regions. And first, maybe in a homeostatic situation to lay the groundwork. Okay, and especially with a focus on macrophages, because that's the cell type we're mostly interested in. So to do this, we relied on single cell analysis, and in the first instance on droplet-based single cell analysis, we used the 10x system. And let's, for example, start with the whole brain. So if you dissociate the whole brain, you fax sort the CD45 immune cells, and you perform 10x genomics sequencing, followed by a t-SNE projection and phenograph-based clustering. This is typically what you see. So the majority of immune cells in the whole brain are macroglia, as we expect. But there are also clearly other subsets of immune cells, such as various lymphocytes, B cells, T cells, NK cells, and a range of myeloid cells. Okay. Now we expect actually the majority of these non-macroglial cells to sit within the border regions, and so to actually be able to more easily study that, we thought, well, it's maybe better to dissect these regions and perform single cell analysis on them separately. So we did this for the enriched subdural meninges, for example. And what we mean with enriched subdural meninges, well, in mice we were unable to really peel off the pia mater from the brain. So what we did is we took a very thin slice from the dorsal cortex, of which the dura mater was removed, to enrich for the pia and the arachnoid mater. Okay. But there is still some contaminating cortex there, so you have to keep that in mind. We also collected the dura mater from the dorsal surface, and also the choroid plexus from the lateral and the fourth ventricles. Now, what you immediately see is that there's really a lot of immune cell diversity in these border regions, especially, for example, in the dura mater, right? So, for example, we really had nice populations of dendritic cells, some conventional dendritic cells, CDC2, subsets CDC1. We had plasmacytoid DCs. We even had an interesting population of dendritic cells which had a real migratory gene signature. So a gene signature that you also find back in migratory DCs in draining lymph nodes, for example. And also a large fraction of macrophages, and these are shown in this reddish color, and we actually call them BAMs, or border-associated macrophages, which is a term that was introduced by the Becker lab. Okay, so for example, we have a large population of BAMs in the choroid plexus, which is the major subset. We have a nice subset of BAMs in the dura, and also here in the subdural meninges. Okay, together with these contaminating macroglia from the cortex. Now, what you could also do, you can pool all of these data sets together, and then you get this beautiful immune map of the homeostatic immune compartment of the brain, where now even additional subsets which were not easily visible in the separate data sets become visible, such as innate lymphoid cells, gamma delta T cells, and so on. And in our manuscript, we actually provide a whole list of signature genes that you can use to identify these various cell types. And I also like to tell you that if you're interested in actually exploring our data sets, all of them are put on brainimmuneatlas.org, together with quite a user-friendly tool to explore your gene of interest or check its expression in any of our data sets. So make sure to use that if you're interested. Now, as I said, we're mostly focusing on macrophages. And for example, you can here nicely see C1QA is nicely delineating all of these macrophages. And so we have multiple BAM subsets in addition to microglia. Now, what was interesting is that you actually see that these BAMs are segregating depending on the tissue origin, right? So this in green here, we have BAMs coming from the choroid plexus, from the dura, and in purple here from the subdural meninges, okay? So this was already indicating that perhaps there is some tissue-specific signatures that these BAMs show quite some diversity depending on where they originate from. Now, to look into a bit more detail, we selected all the macrophages and then reclustered them. And then you get these brain macrophage map, which shows that there are at least, besides of microglia, there are at least 6 other main BAM subsets, okay? So let's move a bit through them. So in the subdural meninges, you mostly have this green cluster here, which we call the SD, or the subdural BAMs. While in the dura mater, you have these two subsets, these two main subsets, which we call the D low BAM in red, the D high BAM in blue. And the low and the high actually this nomenclature refers to MHC class 2 expression, okay? And this will come back later. We also have a small number of these D high BAMs actually in the subdura, okay? The choroid plexus is also, again, mainly consisting of two main populations, the CP low BAM and the CP high BAM. Again, the low and the high refers to MHC class 2 expression. And now we have this interesting peculiar population, a small subset, which we call the CP epi BAM. You will later on see why we call it epi. The epi stands for epiplexus. And this cluster, this subset clusters separately from microglia and from the other BAM subsets. Okay. Now, what is interesting is that these BAMs really had distinctive gene signatures. And we try to actually visualize this in a way to reveal common themes, common modules. And I think this worked out quite nicely with this heat map, where you see the expression in all of the 6 different BAM subsets together with expression in microglia. Okay, and so this shows that you have, for example, genes that are upregulated in all BAMs or are high in all BAMs, but are quite low in microglia. So these examples are ApoA, MS4A7, LIS2. So these are universal BAM-enriched genes. Okay, but also you have genes that are more specific for certain subsets of BAMs. Okay, for example, genes such as the mannose receptor, CD163, they're enriched in the class 2 low BAMs, both from the meninges and from the choroid plexus. On the other hand, you have genes that are restricted to meningeal class 2 low BAMs, such as CLEC10A, CD29F, FOLR2, and so on. So I will not go through the list, but it's just to show you that there's quite some diversity depending on the subset and the region. And you, for example, even have genes that are restricted or highly upregulated in choroid plexus BAMs or in a specific dural BAM subset and so on. So I think this is interesting. This shows that there is quite some diversity. We also confirm this with bulk RNA sequencing, again showing that depending on the border regions, there's quite some differences in these BAM subsets. Now another question is, can we actually translate this to the protein level? And can we come up with, for example, a flow cytometry panel with which we could also identify these various subsets, which would make it much easier, because while single cell analysis or sequencing is quite powerful approach. It's also labor intensive and very expensive. So we tried to do that. So we looked for all of these gene signatures, tried to scan which ones are expressed on the membrane, which ones have antibodies that work, and try to come up with a high-dimensional cytometry panel. And this is flow cytometry, so not mass cytometry. And I think we succeeded pretty well. So first we generated a general panel with which you can identify the various immune cell populations in the brain, but you couldn't distinguish BAMs from the different border regions, okay? So we also tried to come up with a macrophage-specific panel with which we could, in theory, starting from a whole brain, identify the BAMs coming from the distinct border regions. And I think this worked out pretty well. So to give you an example, so here we isolated once more enriched subdural meninges, dura mater, and choroid plexus, and did flow cytometry using our 17-color macrophage panel, and then combined these data sets, right? And here it's gated on the macrophages. So all the cells you see are only macrophages. So, and actually this shows, so this Disney projection shows that cells from the subdural meninges in green segregate from the dura, which is in orange, and which also segregate from the choroid plexus. So this shows that there is enough in this panel, there's enough information to actually distinguish macrophages coming from the various border regions, which is exactly what we wanted to achieve. Now we also try to combine this with a manual gating strategy, with which we could then actually identify all of the different BAM subsets. Okay, and I will not go into the details, but actually this also worked quite well. So we can indeed identify subdural BAMs, the various dural BAM subsets, and choroid plexus BAM subsets. And actually, what you can do is you can then, if you then now map these onto the Disney, you indeed see that it's fairly well working. So indeed, we have here in gold the microglia. We have here in green again the subdural BAMs, and then the various BAM subsets from the dura and the choroid plexus, in addition to the CPAP BAMs. So it's of course the manual gating is never perfect. You have some cells that are or wrongly annotated. But the advantage of a manual system is that you can now actually cell sort these populations or fax sort them and perform additional experiments, right? You can do now bulk RNA sequencing, or you can do functional assays in vitro and so on. Okay, then another thing that was interesting is when you actually now start to look at microglial signature genes in these various macrophage subsets, in these brain macrophages. Okay, so these microglial signature genes were identified previously by comparing microglia with other peripheral macrophages, right, in other peripheral organs. But if you now compare them actually within brain macrophages, you see that not all of them are actually really specific for microglia. For example, genes such as FcRLS are highly expressed in several other brain macrophage subsets, and also other genes such as TREM2, HEXB, and some others show intermediate levels of expression in various BAMs. Okay. Now there are still certain BAMs, certain signature genes that are really macroglia specific, and this is, for example, SAL1, which is a really good example. Others are SPARC or the solute carrier SLC2A5 and so on. But of course, you may now notice that actually I'm not really telling the correct story, because there is always this one pinkish BAM subset which does express all of these signature genes as well, albeit often to a lower level. Okay. So this was actually quite interesting to us, because it was suggesting that this might be a population that is resembling, in a way, microglia. It is not identical, right? Because already you see that the expression of the signature genes is lower, but it has all of these really distinctive genes, even SAL1. Okay, so we thought, well, this is actually a very interesting population, and let's really focus and so try to track them in the choroid plexus. And to do this, we relied on SAL1, which again is expressed in microglia. It's not expressed in any of the other BAMs except for the CPAP BAM. Okay, the expression levels are still decent enough. So what we did is we used the SAL1 CRE-RT2 fate mapper crossed to the ROSA YFP reporter. And in principle, if you inject tamoxifen in these animals, you would get YFP expression in parenchymal microglia. This was already also nicely shown by the Melanie Greater group. And if you would really have a SAL1 positive population in the choroid plexus, you would then also see YFP expression there. And so this is what we did, tamoxifen injections and then isolating the choroid plexus, again from the lateral and fourth ventricle. And interestingly, indeed, when you gate on macrophages from from the choroid plexus, you indeed see that a fraction is now YFP labeled. And this is around 5% in the lateral ventricle, a bit lower in the fourth ventricle, but these numbers actually quite nicely correspond with what we saw in the single cell analysis. Now, you can still, of course, argue, well, maybe the dissection was not ideal, maybe you had some contaminating parenchyma and so on. So it's the real important experiment here was to do the spatial imaging, right? And so this is what we did. So these are now confocal images of the lateral ventricle, of the third and the fourth ventricle, where we stain for EBA1 in red to label macrophages. We have the YFP, right, coming from the SAL1 fate mapping. And then we have cytokeratin in blue, which labels the choroid plexus epithelium. And then this shows something really interesting. So the easiest way is to see it, for example, in the fourth ventricle. So here, again, you see the blue is actually delineating this monolayer of epithelial cells. In between it, you have the stroma, where you have these stromal macrophages sitting there. But they're always EBA1 single positive. They don't show any YFP expression. When we see a double positive, a EBA1 YFP double positive cell, it was always on the apical side of the choroid plexus epithelium. For example, if you can see it here, you can see it here. So this means they're actually really sitting they're bathing in the cerebrospinal fluid. So they're sitting at the outside of the epithelium. The same thing was, for example, seen in the lateral ventricle. This is an example. This is actually a very nice one, also shows ramifications. And again, the ones that are in the stroma, they do not express YFP. Okay, so this brings us actually then to this system, this model, so that the CPAP BAMs are actually now located here at the outside on the apical surface of the choroid plexus epithelium. Right, and the interesting thing is that they're behind a barrier, because again, remember these epithelial cells, they have these tight junctions, and so this is behind a barrier, while the stromal macrophages, right, they are in contact with the blood. Okay, and we think that actually these CPAP BAMs are the Culmer's epiplexus cells, which were discovered decades ago via electron microscopy, where indeed it was observed that you have this macrophage-like phenotype, quite a rare cell, which is indeed sitting on this choroid plexus epithelium. It's quite intriguing when you see them, and bathing in the CSF. But there actually not much was known about these cells. Okay, and so now we propose, so indeed, that these are Culmer's epiplexus cells, and that they seem to resemble microglia, right? But what does this mean? In what way do they resemble microglia? So this is then the next step to really compare the CPAP BAM with parenchymal microglia, and again, some interesting observations were made in this respect. So, for example, if you look at this volcano plot, you see all the genes that are downregulated on this side, on the left side, and the genes that are upregulated in CPAP BAM are on the right side. And to start, indeed, again, you see that the downregulated genes are the homeostatic microglial signature genes, okay, such as TMEM, P2RY12, and so on. They are expressed in CPAP BAM, but they're downregulated as compared to parenchymal microglia. Now, a whole range of other genes are upregulated, and these included genes such as CST7, CLEC7A, ApoA, LPL, and so on. So really a lot of genes that are involved in phagocytosis and lipid metabolism and so on. Now, what was really interesting when we saw this data, we immediately thought of the paper that had recently came out at that point from the Ido Amit group, a very influential paper on the DAMs, or the disease-associated microglia, that were observed in amyloid models of Alzheimer's disease.
Can you just maybe share the screen, because I think at a certain point, just in the last slide, I think we lost the...
Oh yeah, I see. Okay, I see.
And there is a question in the meantime.
So did you find these co-reflex macrophages circulating in the CSF, or you only find them in the
yeah, okay. So whether we find them circulating in the CSF? That's what you asked? Yeah, exactly. So so I have to say we haven't actually performed single cell on the CSF, because that's really very difficult, because you have so little amount of CSF from a mouse, and so little amounts of cells are in there in naive mice. But we've done some FACS, and actually we don't seem to find them back. So at the moment my guess is no, they're not in the CSF. But this is not 100% sure, so we need to confirm this. Okay?
Yes, thank you.
Now I will share them again. But what is actually interesting is that there do seem to be, I mean, this is preliminary, maybe I'm not, better not tell this, but anyway, in the CSF there seems to be macrophage-like cells. So textbook is always saying that in the CSF you have these strictly monocyte or like monocytic precursors. But we seem to see like that there is even some more macrophage-like cells in the CSF. But again, very, very preliminary. But no CPAPs. Okay, anyway, so there was this paper by the Ido Amit group with the DAMs, where they looked at the amyloid models of Alzheimer's disease, and saw that you have a subset of microglia which is showing very similar gene signatures. Okay, so again, these same genes that are involved in phagocytosis and metabolism are upregulated. So this was quite striking, and we thought, well, let's actually compare these two subsets, and let's use it using the same single cell pipeline, and let's try to assess this in a new Alzheimer model. So we used the APP-PS1 this time, and isolated immune cells from the whole brain, so CD45 immune cells, and performed single cell sequencing. This indeed showed that you indeed nicely find only in the IPP mice a new subset of microglia appearing, these DAMs, very clearly. For example, if you look at expression of CST7, a good signature gene, you see it's clearly expressed there, and you see a downregulation of microglial signature genes. Okay, and this subset actually increases as the mice are aging, which we also show. And then it really confirms everything that was shown by the Ido Amit group. And if you now compare the two subsets, so if you compare DAM versus microglia, or you compare CPAP-BAM versus microglia, you really see that very similar genes are upregulated in the two populations, and very similar genes are downregulated. Okay. And if you now compare this on a scatter plot, and then here we now show in the x-axis the differential expression log p-value of CPAP-BAM versus microglia, so meaning all the genes on the right side are upregulated, on the left sides are downregulated, and the same for the y-axis, but then for DAMs. You see that all the genes here within this red rectangle are also the genes that are shared, so that are upregulated in both subsets, and it includes most of the relevant genes that are found in DAMs. Now, they're not identical. So, for example, the dams showed an upregulation of TREM2, which is, of course, very relevant. There is no upregulation of TREM2 in CPAP BAMs. They do express quite high levels, or similar, but it's similar level to parenchymal microglia, so it's not upregulated. And there are genes that are only seen to be upregulated in the CPAP BAMs, such as, for example, the transthyretin, which was found in all CP BAMs. Okay. So anyway, this is showing that these epiplexus microglia, or the CPAP BAMs, they seem to share a core transcriptional signature with DAMs, with macroglia that are observed in conditions of neurodegeneration. Okay. Now, what this actually means, I will discuss later, or at least what we think this means. We'll discuss it later, because first I want to go into the ontogeny a bit, because we actually think that this is not just a subset that is resembling microglia. We actually think that it's really a bona fide microglia, okay? So that it's just a non-parenchymal microglia. But to be able to prove this, we did some ontogeny studies. And in the first instance, we used the 633CR1-CRE-RT2 and the FLT3-CRE fate mappers, right? A bit as was done by Marco Prince already. And this also nicely confirmed the data that was already obtained by Marco. So indeed, showing that subdural macrophages, they don't seem to get bone marrow input, so that they self-renew in situ. In adults at least, and that in the choroid plexus you do get bone marrow contribution, right? And so that's a mixed population, partly embryonically derived, partly monocyte derived. Now we also analyzed the dura mater, which was not assessed previously, and so there we saw that both of these macrophage subsets in the dura actually are progressively replaced by monocytes. And especially the D high, so the MHC2 high dural macrophages, they are completely bone marrow derived at around 8 to 10 weeks of age. Okay, but the CPAP BAMs, they don't receive any bone marrow contribution. So in for example in the FLT3 animals, they don't incorporate any YFP reporter expression. So they seem to be really embryonically. But of course, this doesn't necessarily mean that they are always strictly coming from SAL1 precursors, right? So we can still imagine that, for example, SAL1 negative stromal embryonic choroid plexus macrophages may once in a while traverse the barrier, get into a new location on the apical side of the choroid plexus epithelium, and then start to upregulate microglial genes, start to a bit resemble microglial genes because of the environmental signals that are encountered. Okay, so to investigate that, and also to actually assess what is happening when you actually push the system a bit. For example, if you would open up the niche, if you would deplete these macrophages and allow them to repopulate, does it still happen internally, or do you now force bone marrow engraftment? But to answer this question, we first needed to actually check whether you can deplete these BAMs, and we wanted to try it via the CSF1R inhibition, which was shown previously by the Green Lab and by many others now, that it really works very easily very nicely to deplete macroglia. So we used the Plexicon, the PLX chow, and allowed mice to sit on chow for 21 days or on control chow. And this showed that indeed, as was shown previously, this nicely depletes parenchymal microglia. Right, you can nicely see it here. But also it really nicely depletes all of the BAM subsets, okay? So you get a really strong reduction in BAMs. Often this seems to be more efficient than the macroglia even, okay? Now, if you allow these BAMs to repopulate during 10 days, you see that for example, microglia are repopulated, which was known, but also the other BAMs. So the BAMs are also all repopulate within 10 days. Now, an exception was actually the subdural BAMs, which after 10 days were not repopulating, and this may indicate, this may indicate that they actually have a limited self-renewal capacity. Okay, but we never went longer than 10 days, so it isn't clear yet whether this is something that is rebounds later on or not. But it is a striking observation. Now, can we actually, so we can deplete the bumps, they repopulate within 10 days. So can we now actually combine this with fate mapping and to see whether after depletion there is a repopulation from an internal pool or from a bone marrow engraftment? So to do this, this is schematically how this would work. So you start, for example, with the CH3CR1 CRE-RT2 YFP strain. If you would inject tamoxifen, you get YFP expression in all of the brain macrophages, right? But also in blood monocytes. What has been shown previously by Stephen Jung and Prince Labs, if you wait several weeks, you lose your reporter expression in monocytes, while this is retained in your brain macrophages, okay? So this is then the time point where we start to give the PLX to deplete all these macrophages, and then we allow them to repopulate. And now, so if they would repopulate from an internal pool, you get YFP expressing cells that will fill in the niche. If they're coming from monocytes, then you will have no reporter expression in these subsets. And so the percentage of YFP would then drop within these populations. So this is then what you actually observe. So you see for microglia, there is no drop in YFP. So when they repopulate, the YFP levels are exactly the same. So this means that they strictly repopulate from an internal pool. This was already shown previously. And the same one now was observed for CPAP bumps. Again, showing so there is absolutely no bone marrow contribution, or at least it is fully repopulating internally after depletion. Okay, but what was interesting is that all the other bump subsets did receive bone marrow input, even the subdural bumps. So at least to some degree. Okay. But what is also important here is that you realize that even though we had almost completely depleted the YFP macrophages, still after repopulation, a considerable fraction, especially for example in the D-low BAMs or in the CP BAMs, were still YFP positive. So this is showing that the few numbers of YFP macrophages that remained, they were actually able to very efficiently repopulate. And so while there is an engraftment happening from the bone marrow, still internally also these macrophages were quite efficient in filling up the niche. So I think this would suggest that perhaps choroid plexus and perhaps also the D-low BAMs will always be of a mixed ontogeny, so that you always will retain a fraction of embryonic macrophages, even after inflammation and cell death so depletion of macrophages and a repopulation. But of course, these YFP expressing macrophages here are not necessarily embryonic, so this is not proving that point. So and finally, then what was actually also important was to repeat this experiment with SAL1 CRE-RT2 system, and this showed, so first of all, again, to show you, SAL1 is only expressed in macroglia and CPAP BAMs, and indeed you don't have any reporter expression in all the other BAM subsets. But in the CPAP BAMs, upon depletion and repopulation, all the CPAP BAMs retained their YFP reporter expression. So this shows that they strictly repopulate from a SAL1 precursor. Okay, so to conclude, so these CPAP BAMs, they are never derived from SAL1 negative stromal macrophages. Otherwise, they would have lost their reporter expression. So it means that they're either repopulating internally, so that they are self-proliferating, that they're proliferating, and the few remaining CPAP BAMs then fill up the niche again. Or, which would be a more exotic explanation, is that parenchymal microglia may actually move from the subependymal space onto the choroid plexus epithelium. So it would really migrate from the parenchyma onto the choroid plexus. If this would be true, this would show an unprecedented level of motility for microglia. And this may even actually happen under homeostasis as well, right? Maybe this is the way that these cells are replaced. At the moment, we don't know, right? We don't have the tools. We don't have any fate mapper that can actually distinguish between microglia and CPA epibalms. But I think this is really an interesting question to look at. And so the conclusion is that actually we do think that these CPA epibalms are a true microglia, so a bona fide embryonically derived microglial subset, but that they're inhabiting a non-parenchymal niche. But so what does it mean now that they have this shared signature with the DAMs? So these are macrophages that are present in healthy young mice. So there's not a disease involved. So we think that this shows that actually DAMs are not necessarily a disease-induced phenotype, but that it may actually reveal a second functional state of microglia. Right? To give you an example, so microglia that are sitting in the parenchyma, in the healthy parenchyma, their main role is to actually support neurons, to help in synapse remodeling and so on. Okay, and for that they have a transcriptional program, and that's the homeostatic gene signatures that we see. But once things go wrong, for example, once you get cell death, you get inflammation and disease, they need to shift gears, need to adopt a new functional state to cope with these insults, right? And this is what you see in the parenchyma with DAMs, and this is also what you see in the CPA epibalms, because they're now sitting in a new environment. They don't have any, there are no neurons, it's a neuron-free environment, so they adopt this new functional state. Now, what they're actually doing in this environment, we don't know, right? And this is something we're really interested in to try to understand. Okay, it's time. Okay, then a final part I would like to discuss is actually what are the transcription factors or the gene regulatory networks that are actually driving macrophage diversity in the brain. And to investigate this, you can actually start with your single cell data and use some of these very interesting pipelines that have been developed. For example, the scenic algorithm that is developed by the Stannard groups, which is actually allowing you to look for main transcription factors and gene regulatory networks that could explain the transcriptional activation of your cell types. Now, what scenic does is actually it scores. First, it starts with a co-expression matrix, so it looks for genes that are co-expressed with a particular transcription factor. In the next step, it then actually removes all the genes that do not have a motive for that transcription factor in its promoter regions. Okay, and then you end up with a regulon or a gene regulatory network. And then in the final step, it actually it scores the activity of the entire regulon in every individual cell, right? So it looks for the activation of all the genes that are in this network. And so this means that also this regulon approach is more, it's less sensitive to gene dropouts. Now what you can do, so here we had t-SNE based on gene expression. You can also have a t-SNE based on regulon activity, right, which is shown here. And here the colors actually match the colors of the transcription-based clustering. Now it shows actually that it's quite nicely recapitulates what we saw previously. So here we have the microglia. Here we have CPA epibalms, which cluster separately from microglia and also separately from the other BAMs. But in this case, they're closer to microglia, actually. And you also have these various other BAM subsets, such as SD BAMs and the DLO BAMs, and the various CP BAMs. Okay, so it's not as clear as with the expression, but actually it quite nicely correlates. And this also gives you a list of potential transcription factors or regulons that may be important in obtaining this activation that you see. Okay, and of course the next step is then to confirm this, right? This is a great starting point to try to dissect the gene regulatory networks at work, but you have to confirm this. And we tried this with one of the transcription factors we were interested in, namely IRF8, which showed a high regulon activity in microglia, but a reduced activation in CP-AP BAMs. So it was already suggesting that perhaps the shift from microglia to CP-AP BAMs, that IRF8 may have a role there. It was quite low in meningeal macrophages, and there was low into intermediate levels of regulon activity in choroid plexus BAMs. Okay, so what we did is we conditionally knocked out IRF8 from macrophages using the FCGR1 or the CD64 Cree mice crossed with IRF8 flox animals, and performed single cell analysis on whole brains or on the choroid plexus of these animals. Now this showed that actually when you here, for example, make an aggregate of the microglia and the macrophages from these data sets, from the wild type and the IRF8 knockouts, you actually see that the wild type microglia and the IRF8 knockout microglia completely segregate. Okay, so showing that there is quite a big effect. And indeed, if you then look at the genes that are differential, you, for example, see that there's almost a complete loss of cell 1 in these IRF8 knockout microglia, right? Which is quite striking. You also see a very strong reduction in other signature genes such as P2RY12 or SPARC, while on the other hand, genes such as ApoE or other BAM-related genes are upregulated. Okay, so genes such as MS4A7, PF4, which were more BAM signature genes, are now induced, and often to a similar level as to choroid plexus BAMs. So this is showing that in the absence of IRF8, these microglia seem to adopt a more BAM-like phenotype. Okay, which is interesting. But they also have genes that are more related to CP-AP BAMs or DAMs, such as CST7 or CLEC7A and so on. So again, this may indicate that indeed the CP-AP BAM signature is partly regulated by IRF8. Okay. Now, also choroid plexus BAMs showed quite a strong difference in these IRF8 knockout animals. So, for example, here we have the BAMs coming from wild type mice. So these are the CP high BAMs. Here we have the CP low BAMs. And here are the knockout counterparts. Okay, so one of the first things that you see is actually that you have lost the majority of your CP high BAMs in these knockout animals. So the main subset that remains are the CP low BAMs, but they're also different from the CP low BAMs in wild types. So now they start to, for example, express genes such as CLEC4N or CFP that are not found in normal CP low BAMs. Okay, and these are actually genes that you do find on meningeal BAMs. Okay, these were genes that were actually quite specific for meningeal BAMs, and genes that were specific for CP BAMs, such as, for example, LILRA5, it's actually reduced in these knockout CP BAMs. So the idea is that it seems that these CP BAMs now actually from the knockout animals start to resemble a bit more meningeal BAMs. Okay, and now we can think of a model where, for example, you would have something like a protobrain macrophage, right? Which is most similar to the meningeal BAMs, but that due to IRF8 skewing, you get then actually differentiation towards adult CP BAMs and towards adult macroglia. Okay, but it's just shows that IRF8 is crucial for the transcriptional identity of microglia and CP BAMs. Okay, so this is the final slide, again an overview slide. So I think our work together with others that came out around the same time has now really led to a nice framework for understanding brain macrophages. And I think the next big step is now to really start to elucidate their functions both in homeostasis and in disease. But I think we have a good framework to start with that. And then the final slide is really important to actually thank the people that did this work. So the majority of the work you saw was done by a very talented PhD student in my group, Hannah van Hove. We're a small team, but very cooperative. So actually everybody helped. So also Isabel, Karen, and Anna. Also important collaborators from VIB Center of Inflammation Research are Johan Jindrachter and Ivan Sijs, and especially Lisbeth Martens from the Ivan Sijs group, who was really instrumental for a lot of the bio-IT analysis. And of course, a lot of the funding agencies are acknowledged. Okay, thanks a lot.
I think the live audience are quite happy. Since no one wrote any questions yet on the Q&A section, maybe we take some questions from the live audience. Or any? In the back.
You said at the end that you still have the question comparing to what they do.
Yeah.
The different
bands. Okay, so I will rephrase the question again. He's just wondering if you have done any experiments to elucidate on the functions of the BAM populations in the choroid plexus, or the microglia-like population, the FU BAM.
So not really. So that's something that's ongoing, but the problem is that we actually need new tools to be able to really target specific subsets of these BAMs, right? If you would, for example, if you want to study the function of these CP-AP BAMs, it would be useful to have a tool with which we can actually genetically target them. And at the moment, that's still something that's lacking. We are thinking of in the meantime, of alternative approaches to study this, but at the moment
can't you inject
it's too early to
the
tell
theratoxin into intraventicular, so that way if you have like a cell free with the theratoxin detector, it doesn't affect microglia necessarily, but can affect the ones that are swimming.
Yeah, that's a good remark. It's a good remark. So we tried that actually, but it doesn't really work that well. So we don't really know why. So if you inject, we inject actually via the cisterna magna, because I don't like to do ICV injection because that induces quite a lot of inflammation and damage, which we actually see quite a lot of monocyte influx and really a lot of things happening. So I'm not a big fan of that, but we tried ICV injection, cisterna magna injections. But it's hard to deplete them actually. And we also do this in an inflammation model. And again, we don't really know why, but even injecting quite high doses of diphtheria toxin is still not depleting these cells. What we now are trying is if you inject systemically, but try to alter your dose to find the optimal dose with which you perhaps would mainly deplete the microglia around your ventricles and less around.
Okay, we have another question. I think it's also quite related. So they were wondering if you checked for the presence of the choroid plexus epi-BAMs in a disease context, whether or not something happened to this population in regard to functionality or cell density or so on.
Yeah. So that's actually very interesting, and I will ask you to come to Berlin, right, in next year, and there I will show some really exciting data regarding this. Okay, so I will share that.
I have a short
Berlin,
question.
Okay.
It's not regarding the BAMs, but regarding the microglia actually. So you showed them as one single cloud, but do you think that actually there is, or several people have argued that microglia are heterogeneous? So what could be your comment on that?
Yeah, sure. So we also actually stated in the manuscript that we we pooled macroglial subsets, so that we just artificially made it one cluster, because we're mainly focusing in this manuscript on the BAMs. So I do think there is macroglial heterogeneity, but it's not that clear. Okay, so my take is that macroglial subsets are typically small populations that are in quite unique environments in the brain, such as, for example, the CPAP bumps. But even in the rest of the brain, I think you will find clear heterogeneity. But in an adult brain, it's not easy to find, because the majority are actually just homeostatic parenchymal microglia. Whether there is really a lot of difference between, for example, cerebellum macroglia and cortical macroglia, I don't really know. I don't have based on the single cell data, the differences are not that big. But of course, this doesn't say that functionally they may be quite different. So maybe a few genes difference may still have a big functional impact, right? But you will not easily pick that up.
And I think I also maybe have another last question. What do you think is actually different in the environment for the choroidal, so that the epi BAMs, for example, that is similar to the neurodegenerative context? Is it rather the absence of some signal from the neurons, or is there something specific in the CSF that mimics the disease context?
Yeah, I think it could be a combination of both. So I think indeed, if you don't have the neurons present, if you don't have the normal environmental stimuli that you have in the parenchyma, this will already have some dramatic effect on the macrophage, right? So the niche is really important in shaping, I think, the macrophage transcriptome. But in addition to that, indeed, you have a lot of additional molecules in the CSF, right? You could have amyloid in the CSF. You have perhaps lipids or other metabolites that shape the phenotype of these macrophages. Yeah. For the moment, we don't really know what is the main driver of the phenotype.
Okay, great. I think it's time to conclude the session, and I would really like to warmly thank you, Kia, for giving us your time, and we're all looking forward to seeing you again in the symposium, and looking forward to seeing more of your research and highlighting again the APMs.
Thanks a lot for having me, and I'm also looking forward to seeing you all back in Berlin. Okay.
Bye bye.